Good morning, everyone. I'm Pranali, your host for today. I welcome each and everyone for today's Sadhguru Summit. This is our 290th virtual webinar. Topic for today is Biocompatibility Evaluation of Medical Devices, which shall be taken by Mr. Viral Sheth. Agenda for the Sadhguru Summit. Particulars, virtual inauguration by Dr. Sundar Kataria, invoking the blessing of Deity chanting of Gayatri Mantra, introduction of speakers, presentation by Mr. Viral Sheth, question and answer session, closing session. Dr. Sundar Kataria, Chairman and Managing Director of ICS Group Companies, Sadhguru Sangh conceived and led by Dr. Sundar Kataria, driven with a mission to elevate the quality, safety and environmental awareness levels of industry. Sadhguru Sangh is a series of educative, informative events aiming to illuminate professionals on possible solutions to current issues of industry and economy. Dr. Sundar Kataria is the Chairman and Managing Director of International Certification Services Private Limited, Academic Qualification, Mechanical Engineer AIM, Honorary Doctorate, Business Management, Experience, Nuclear RAP. He started his career in building India's first nuclear power plant at Rawatbhatta, Kota, Rajasthan. Oil and gas EIL. He contributed in developing India's first oil and gas field at Bombay High since its inception. DNV. He was a head of industry and offshore development with DNV for 2 decades. ICS. He is the founder of a first Indian conformity assessment and inspection body known as International Certification Services Private Limited, which is a provider of a total quality solution under single umbrella with a wide spectrum of services. Achievements and awards, Lifetime Achievement Award of NACE Kokon, and received several national and international awards. Now I request Dr. Sundar Kataria sir to virtually inaugurate today's session.
शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तुते दीपज्योतिर्नमोऽस्तुते शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरु त्वं कल्याणम् आरोग्यं धनसम्पदः.
Please join us for the recitation of the Gayatri Mantra. Introduction of the keynote speaker. It is my great pleasure to introduce today's esteemed speaker, Mr. Viral Sheth, who will be delivering an insightful section on biocompatibility evaluation of medical devices. Mr. Viral Sheth is a highly accomplished regulatory affairs and quality management professional with over 22 years of extensive experience in the medical device, pharmaceutical and microbiology sectors. He currently serves as the Director Operations at Medrec Consultancy LLP, Director of Biotox by Medrec, and Principal Consultant at HL Associates. Academically, Mr. Sheth holds a Master of Science in Microbiology and a Bachelor of Science in Microbiology from Hemachandracharya North Gujarat University, providing him with strong scientific foundation which has supported his remarkable professional journey. Throughout his career, he has gained extensive expertise in Indian medical device regulations, European Union medical device regulation, US FDA and UK medical device regulation. He also provides European authorized representatives, UK responsible person and US agent services through European partners, helping organizations achieve global regulatory compliance. Mr. Sheth has played key leadership roles in a regulatory affairs and quality management, including serving as a regulatory affairs manager at Solco Biomedical and quality assurance manager at NG Plus Private Limited. His professional experience spans quality control, quality assurance, regulatory affairs, business development, consultancy, regulatory documentation compliance, audits, and quality management systems, making him one of the most leading experts in the field. Everyone, please join me in extending warm welcome to Mr. Viral Sheikh. So, the virtual stage is now yours. We look forward to your valuable session.
So, good morning to everyone. Myself, Hiral Seth, having almost 20 years of experience in medical device. So, we are the regulatory consultants, and today's topic is biological evaluation of medical device, that is the biocompatibility evaluation of the medical device. The main objective of today's session is to describe the methodology to be followed for the biocompatibility, irrespective of the orthodox procedures that are we used to previously follow just by identifying the tabular test that is already given in the older version, that is ISO 10993 2018 versions. So people generally used to identify the which test is applicable to my device. So now the new version is there, ISO 10225 version, that is published in November 25 and to be implemented in coming days by the demanded by the regulatory authorities. So it said divided the tables and almost the methodology of the not following directly testing, that is the evaluation biological evaluation of the medical device by chemical characterization and including the other biological effects are already there. But that is too much not required or demanded from the regulatory authorities. Nowadays, the demand of the regulatory requirement regarding the biocompatibility is somewhat very stringent. So they want to follow some systematic approach to reduce the overall animal testing and to reduce, replace or reform these are the three core principles that is followed in the globally or to reduce the animal testing only. So as far as possible, the regulatory agency is asking why are you directly performing the test irrespective of the systemic evaluation that is described in the standards. So today we will discuss and talks on the methodology to be followed to do a biocompatibility evaluation. So today these are the overall index and we will going to deliver the overall description of this context. So as we all know, what is the medical device? So this may be a possible example of the medical device that is subject to a mandatory evaluation under regulatory requirement of the India, Europe, US and anywhere where you are intending to sell your medical device and those comes in contact with direct or indirect contact with the patient. So doing a biological evaluation is the identifying the chemical and physical characteristics of your device that is coming in contact with the biological part of the patients. So how the biological part of the patient as per claim of your intended purpose and indication of your device is suitable to the patient is more important. What are the risks associated with your device? Risk, here we will talk only the biological risk. So what are the risks that is associated with your device is required to be systematically evaluated by following the requirements that is given in ISO 10993 2025. So the current standard that is globally required to be followed is ISO 10993 Part 1 2025 version and older version is 28. So those who had conducted test as per 2080 version does not mean that you again conduct the test as per 2025. The standard talks on the biocompatibility evaluation of the medical device or their parts or components that is coming in contact with the patient. The contact that comes in contact with the patient may be a direct or indirect. For example, catheters that are directly inserted into the human, so they are direct contact. Syringes without needles are indirect contact. Respiratory pathways are indirect contact. Again, you should have knowledge of the duration of exposure of your device. So firstly, we understood the what is medical device, why biocompatibility is required. Then after, you should have knowledge of the contact type of your device. It may be direct or indirect, and depending on the contact nature, you will need to assess some special tests that we will discuss in coming slides. Then after, duration. So duration may be a limited, that is up to the, if your device is coming in contact up to the 24 hours. The next is 1 day, 24 hours to the 30 day, and third one is lifetime of the device, that is longer term, that is more than 1 month. For example, long term in case, for example, implantable device. Those are coming in a lifetime of the device or maybe more than 30 days. For prolonged exposures, we can have example of the catheters, feeding tubes, Ryle's tube. Limited exposure are the syringes, patient monitoring device, diagnostic devices. There is also one part that is the brief tissue contact. That is limited. That is almost less than 60 seconds, less than 1 minute. The ISO standard also talks about the biocompatibility evaluation of the device that are coming in contact up to the only less than 1 minute. For example, blood lancet, hypodermic needles. So these are the possible contact duration of your device maybe, and this all information is required to evaluate your device systematically. So before starting and going in deep, let us understand the definition that is given in the 2025 version. So what is biocompatibility? It's the ability of our device or material to perform with appropriate host, means patient response in a specific applications. Specific applications means claimed by the manufacturer for particular medical device in particular clinical conditions. So it talks to understood what are the possible biological effect depending on the specific application of your device required to be evaluated. Biological effects are the observable and measurable response of cells or a living organism to a physical characteristics. Here you should understand, and they are mentioned the physical characteristics and chemical constitute of the device. So biological effects depending on the physical and chemical parameter of your devices, including physical agent if something is additionally used in your device. That may be animal derived parts or animal tissues or nanomaterials. The biocompatibility evolution itself is biological evolution. It's an integration of the risk management process in medical device. The risk management is required to be integrated or followed as per ISO 14971 versions, and ISO 24971 is the guidance document for the implementation of the risk management. So integration of risk management in biological evaluation to identify, evaluate, and control biological risks are known as the biocompatibility evaluation or biological evaluation of the medical device or material or parts of material. Chemical characterization is the demanding word, and most of the regulatory authorities are asking and even enforcing manufacturers to follow the chemical characterizations. It's a process of obtaining chemical information of a device or material through information gathering or generation, such as literature review and testing. So separate parts are there. If you refer the biological effect and a definition, so it talks on the physical characteristics. So to understand the physical character of device, you should follow the ISO 10993 part 19. For chemical constitute, we need to follow the part 18 of this series. So it's a series of standard. It's not a standalone. There ISO 10993 part 1. So there is almost more than 25 to 30 different series of these guidance documents are available. So depending on the nature of the our devices, we need to follow the different types of the parts of that device. So once you go for the chemical characterization, it talks on the extrables and leachables. Leachables are that is something can be released from your device in clinical condition. Means as per the label claim of our device intended by the manufacturer. Extractable means what can be extracted. So leachables is clinical conditions and extractables are the using of the different laboratory solutions to extract something in acylated mode using different chemicals that is recommended in ISO 10993 part 12. So the new standard also talks on the identify the foreseeable misuse of the your device. Though you are claiming your device for the use up to 24 hours. But the clinical conditions and hospital healthcare scenarios is like that. Suppose the infusion set, most of the manufacturers are claiming the for the single use and for up to the 24 hours only. But when we go to the hospitals, you can see and the same IV set is being used for the multiple times. So they don't have times and they cannot replace the IV set periodically as per instruction that is given on the IFU. So in generally practice that I had seen an IV set is mainly used up to the 3 to 4 days or up to in some cases up to week also. So though it is claimed for the single use up to 24 hours in healthcare sectors, it is the normal human natures and so they are extending the use of your device. So it's a possible misuse of device as per claim, but during evaluation, you should also understood this behavior of the healthcare industry. So as I said, these are the different parts other than ISO 10993 part 1. Part 1 is the generic versions required to be understood and it describes the risk methodology to be followed for the biocompatibility evaluation. What are the minimum requirements of the biocompatibility plan and reports? What are the strategy to be followed? Part 2 talks on the animal welfare requirements. And so it's a responsibility of the clinical research organizations or third-party testing laboratory to ensure the animal requirements that is being recommended in part 2 that we will also discuss on that in coming slides. So these are the different chapters. So depending on the nature of our device, we should ideally understood which part is mainly applicable to me as per the intended contact and contact duration of the devices. So 10993 part 1 25 version is last implemented in November 25. So rather than directly going analysis, if you refer the section 6.6.2, it gives the idea of the gap analysis. So those who are already in the existing market and those who had already conducted the biocompatibility test, they must do a gap analysis to align with the latest requirement of already on the market and established an acceptable safety profile. Instead of the historical information shall be reviewed and documented to determine whether any differences medical device were used in prior review, additional biological effect, related literatures and adverse event will impact the conclusion of the biological safety. So it means that though we have conducted testing, but there are some questions whether you conducted these testings on the worst case scenarios. Is there any change in the raw materials that is being tested at time? For example, in 2014 or 2016, you conducted some test on the material. Do you have or integrated the information of the manufacturing residue or adjuvants that is being used for the manufacturing? So this all information is required and mandatory under 2025 and they are asking to do a gap analysis. The limitation of The standard is the evaluation. It's written in note the evaluation of this risk related to infectious agents like bacteria, mold, yeast, virus, and TSE agents is not within this box. So when you are talking this some microbiological concerns of your device and animal agents, so it is the beyond the scope of these biological evaluations. It does not also talks on the evaluation bacteriological, that is bacterial endotoxin or that is present on your device. So this is the main limitation of this latest version. So the standard talks to firstly do a biological equivalence of your device by following the chemical and physical equivalence. For chemical, we should refer part 17 and 18. Part 18 is for the chemical characterizations, where the involvement of the toxicologist is required to comply to the requirement of the part 17. So part 17 talks on the toxicological risk assessment. So it's a combined and joint exercise of the part 18 that is being followed at the third party laboratory for the chemical characterization and which requires the involvement of the toxicologist. Whereas looking depending on the nature of the some physical properties, so if that is applicable to your device, you should follow the part 19. So once you confirm the material equivalence by chemical and physical, you can talk on the clinical condition equivalence of your device, and everything is matching to the predicate. Predicate means the device is already approved by concerned authorities and that is available in the market. So you need to do a comparison of your device with the predicate depending on the interest of your regulatory authority, maybe India, Europe, US, and others. So if you follow the structure of the Europe and US, as a consultant, we understood once you comply to the all the requirements of the Europe and USA, you should indirectly comply requirements of the other countries also, irrespective of the administration formalities that is mentioned in their guidance document. So this standard talks to claim the biological equivalence by chemical characterization and physical characterization. So earlier, generally we used to follow the tabular format that is given in the standards, and we directly select the test and asking laboratory to provide the solution for the this type of test that is identified by following the table. So this is a totally different approach. So rather than following that directly testing, you should conduct chemical characterization and involve the toxicological risk. For toxicological risk, you must involve the toxicologist. So the latest standard that is talk at given the four tables and here we merge it and it talks to do a systematic test and required to be identified by following tests depending on the nature of the contact durations and contact types. That is whether they are coming in the contact with the skin, mucosal membrane, implant, other devices that are coming in contact with the tissues other than bloods and device those are coming in contact with the blood circulation system, direct blood circulation system. So from all those tests, the first three tests are the gold standard of the biocompatibility requirements, and generally in all the categories, the cytotoxicity, sensitization, irritation test. In all the categories, it is mandatory to perform those tests, and then after, depending on the nature or outcome of the result, you can evaluate or you can, along with your toxicologist, will evaluate the overall property of your devices. So the standard talks to identify the minimum table. It does not mean when you refer the standards. So there mention the one code that is E. E stand for the evaluations. So directly jumping for the testing, at least you should evaluate your device and you should understand what are the test requirements. When standard is suggesting some test, means those are the possible risk associated with the human during the using those type of medical device. So the standard talks on they repeatedly asking to do a toxicological risk assessment to evaluate the chemical information with respect to the specific biological endpoints such as systemic toxicity. So we had understood what are the minimum tests that is demanded from the standards. So the standard also asks to refer the other biological factors required to be understood for depending on the nature of your devices. So if your device have some component or material or manufacturing informations and manufacturing information means so during manufacturing your device comes in contact with the several other materials that is serves as a catalyst or adjuvants or other manufacturing aids that is may require for the manufacturing device. So it may be a immunotoxic or neurotoxic and it may be toxic to the reproductive. The material itself has some pyrogenicity. So the earlier we had discussed about the limitation of the standard about the bacterial endotoxins and here again so there is a one myth in the market. Okay, the pyrogenicity is the bacterial endotoxin. The word pyrogenicity that is mentioned in standard is relating itself to the material of the medical device only. Whereas the endotoxin may come from manufacturing environment and manufacturing chemicals also. So they are talking on the pyrogenicity of the medical device. If you think if endotoxins are there, so you have to reduce and you have to design your manufacturing process in such a way that the whatever the endotoxins are enter in your medical device, how it can be removed. Standard also talks if your device are brief tissue contact that is less than 1 minute. So you can conduct a normal biological evaluations and you can claim the biocompatibility of your device by doing the systemic evaluation and providing and saying that yes, it's a low contact devices, whatever the materials are used are meeting the requirements of the standards. It also talks on the low risk intact skin contacting medical device. It is mainly applies to the external surface and housing of the equipments, patient examination tables, power cables, and other control such as keyboards. Means those are the indirectly patient or operators are touching the different surface of the instrument. So you should also ensure that those devices are being used in healthcare environments are safe. There may be some other device like they can degrade or absorb during the clinical applications. So separate chapters are there for evaluation of those type of devices. Some materials can be changed during the use. For example, absorbable sutures. So when sutures are implanted, some of the sutures are degraded in the medical device after implantation. So their state of is again going to be changed after implantation. Absorption through skin or mucosal membranes. For example, the endoscopes. In endoscopy procedures, the endoscopes are required to be lubricated for the smooth frictioning. So when you lubricate your catheters and when it goes to inside to the human colon, so chances are there that lubricants may leach from the endoscopes and that goes inside the human. there may be a degradation and particulate matters that can be released from your devices during the clinical use. So we have to identify all the possible hazardous situations and materials that is present in our medical device before conducting the biocompatibility test to define whether any requirement of the other biological test is applicable to me or not. It also talks to identify the requirement of the ADME study that is mainly for the absorption, distribution, metabolism, and excretion. It is generally applies if your material and nature of medical device is something absorbing or degrading and releasing some particles. So this mechanism is required to be understood by following certain standards that is as per ISO 10993 part 16 of this series. If it's a nanomaterial, then you should follow part 22. In all the tests here, the reason that you must ensure the animal welfare. So animal welfare part 2 is for animal welfare and it is applicable to all the tests. Part 12 is the sampling of the devices that is being used for the biocompatibility. That is also general chapter and that is applicable for conducting all the tests. So identifying test, you should also consider the additional biological effect that is required applicable to your device or not. So earlier we talked on the biocompatibility evaluation of the medical device as per ISO 10993 part 25. Now if you refer the scope, that is section 1 of that guidance document, it also talks if your device are relating to the respiratory breathing circuits, means ventilator tubes, T-mount. If your device is in this category, so you must follow the ISO 18562 instead of the other ISO 10993 series. It's a indirect contact of the devices. So if you are following in this category, so there are 4 sections in ISO 18562 required to be followed for the indirect contact and mainly applicable to the breathing gas pathway. So when we refer the ISO 18562, so part 1 is the general chapters, same like ISO 10993 part 1, and whereas part 2, 3 and 4 of ISO 18562 talks on the conducting the different tests. In case of breathing gas pathway, we must conduct a test for the particulate matter, volatile organic substances, and leachable. Now again, there is a one third twist in the biocompatibility evaluation. So it's a general direct contact of the medical device. Then after we talked on the respiratory breathing pathways, and now the third category required to be followed for the biological evaluation is the if your device is something relating to the dentistry of the medical device, dental medical device. So these standards have given the 3 groups to be followed for the dentistry of medical device nature. So depending on the nature, group 1s are the normal tests to be followed as per the guideline of the 19003 along with the additional requirement that is mentioned in the specific section of ISO 7405. So group 1 is relating to that only. So you must conduct test as per ISO standard. However, you should incorporate and understood the requirement of the particular sections of the ISO 7405. Group 2 is directly depending on the requirement of the ISO 10993 series part. So part 1 talks on the additional requirement. Part 3 talks on the requirement of the 10993 series, whereas part 3 talks on the 100% requirement of to be followed for the ISO 7405. So depending on the nature of your device and contact duration, you should understood how the evaluation of the group 1, 2, 3 and how what tests are applicable to you. So once we refer the different regulatories, so one of the most demanding in world is the medical device regulation that is controlled by the European Commission under UMDR 745, and when we refer the Annexure 1 and Section 10.2, so it talks on the device shall be designed, manufactured, and packaged in such a way as to minimize the risk posed by contaminants and residues to patient, taking account to the intended purpose of the device and to the persons involved in the transport, storage, and use of the device. So it also includes the risk relating to the biological. Particular attention shall be paid to the tissue exposed and those contaminants and to the duration and frequency. Again, in section 10.4, they talks on the requirement and to identify whether any device or substance or their materials or any manufacturing process that is being used along with their manufacturing aids or consumables maybe falls under this carcinogenic, mutagenic, and reproductive toxic categories or not. It's known as the CMR substance, that is endocrine, mutagenic, and reproductive toxic substance. So it is required to be followed to understand the overall manufacturing process along with the design of your device. So are we following in this category or not? If you are following, or if your device have this type of category, you must involve the toxicologist for the safety evaluation of your device. So the carcinogens are mainly categorized in class 1A, 1B, and 1AB both. Carcinogens means substances or mixtures that induce cancer. That is something relating that by their presence above the some levels may lead to a patient in cancer disease. So there is a systematic list available on the biological literatures, and there are so many studies that is already happened and something is going on. So depending on the presence of this substance quantity, generally the standard stocks, this substance must be present in your device with the less than 0.1% volume by weight of. So let us take one example. For example, DHP is somewhat carcinogenic that is present in the normal disposable, for example, infusion sets. So the alternative to get the removal of that substance from that, you must manufacture your device that may be DHP free. Again, if you want to manufacture your device with the DHP, that is presence of the phthalate, so you must keep those DHP present. That for means we need to understand the formula of your tubings and drip chambers. So the overall weight, if the overall weight of my device is 14 grams, so then it should be less than the 0.1% of the 14 gram. That is the dioctyl phthalate. So generally in that industry, the plastic tubings are made with the PVC resins and some plasticizers. So DOP is the one of the plasticizers. So either you can replace the other part plasticizers, so you can claim the DHP free. And if still you wants to incorporate those EMR substances, then you must ensure the weight that is recommended in UMDR. So there is a one guidance document uh, published by United Nations in Annexure 3, which talks on the different safety signs to be incorporated in your labeling, that is either instruction for use or labels or surgical technique whatsoever. So if EMR substance are present in your devices, so the user should have knowledge by following the certain universal code that is being followed to the worldwide. So you can see here some numbers P01, P202, P280. Again, if you refer these all numbers to those who had followed the UDAMED structure, and you should find this all similar codes. So these are the universal code that is defined for following certain instructions for the prevention, response, storage, and disposal if something is present in your devices. In Europe, there is also Article 59 or Article 53, that is EC 528 for biocidal and REACH compliance. So you need to refer those articles if EMR substance is present in your devices. I'm just sharing another slide. Wait. So this is the list, and that is the hyperlink that is just open to describe you the how to identify whether my device is having CMR substances and what are the list. So this website gives you idea and also quantity. What are the safe quantity of those substances? So you can identify the list of the CMR substance from here. Just a minute. आप बताएं नहीं मालूम फिर शेयर नहीं कर Are you able to see the list of the CMR substances?
Yes,
Yes,
sir.
sir.
So this is the website and generally gives idea the presence of the whether CMR substance is present in your device or not. So you how you got the information. So you should have the material safety data sheet of your raw materials or manufacturing processing aids. So once you refer the material safety data sheet, it have the CA chemical abstract substance number in your material safety data sheet. So you should search whether my this CAS number falls under CMR substances or not. If something is present, then it is required to be evaluated in serious way and to understand the safety of the patients. So you must refer, and why this is why we require the information of the manufacturing processing aids for the biocompatibility evaluations. For example, in case of the orthopedic implants, people are generally saying, say, we are manufacturing the materials from the implantable grade materials that is already demanded from the regulatory authority. Yes, you all are agree and we agree with you. Whatever the materials that is recommended for the medical implants, but you are not directly selling the device that is directly recommended because you are doing the lots of the manufacturing process that is fabrication, polishings, and blastings, buffings, and laser markings and everything. So you should also understood the risk associated with the manufacturing process. And manufacturing process are not going to directly. You are also adding some coolant agents, some oils, and other chemicals for the performance of your processes. So what about the those chemicals and processing aids that is being used for the manufacturing? Are they removed in sufficient way that leads to the biological safety of your device? So for identifying and getting the answer of those questions and those risks, you must conduct the clinical characterization of your device. So this is also one of the another website where you can find the idea of the CMR substance along with the EC numbers. So this also gives the idea of the how to identify the CMR substance is present in my device or manufacturing techniques. So depending on the presence of the CMR substance and quantity, you must label your device accordingly for the prevention, response, storage, and disposal of your devices. So the biological overall evaluation of device is limited up to the intended use, their purpose, indications, patient populations, and use environment. It is also applicable to the life cycle of your devices. that start from the design concept to the distribution of your device and clinical up to the clinical use and disposable of your devices. So for doing the biological evaluation, you should involve the subject matter expert, that is SME. So what are the minimum risk, or what are the risks to be required to be identified from the different phases of your device? That is, you should identify risk from the raw materials and their compositions and from their manufacturing techniques that is applicable. Maybe your raw material is good, but the manufacturing techniques that is being followed have some biological risk. They are involving substance that leads to some biological issues of the devices. So you must understood or gather all the informations of the manufacturing aids, adjuvants, and their concentrations and contact time. If something is added during the manufacturing process that is required to be removed from your device, so when we are talking that we are removing those adjuvants, so we are doing the cleaning process. So what are the cleaning agents or disinfectant agents being used for your device? And you should also understood the presence of residuals of cleaning and disinfection agent also. How you are removing the clinical agent? How you are removing the disinfection that is being used as a part of process of your device? Some of the sterilization process also raise some biological concerns. For example, earlier we had seen the one chapter that is part 7, which talks on the presence of the residue of the ethylene oxides. So doing a sterilization is a good for that is the removal of the microorganism from the devices. But once you follow the ethylene oxides, so it raises some concerns on the reproduction toxicity to the patients, depending on the presence of the residues that is generated from the sterilant, that is ethylene oxides, in terms of the ethylene chloride and ethylene glycols, and itself a ethylene oxide. So if those residues are present in your device, it is also a toxic to the patient. That also depending on the intended claim, for example, child, adult, everything. So it is required to be assessed whether those residues are in the limit or not. In some cases, the storage condition also leads to a degradation of your devices. So it is also important to understand those aspects of your devices. There may be some packaging contamination, some residues that is or particulate matters generating from the packagings, and those are coming in contact with the medical device. So chances are that the migration of those particles in the medical device and to the patients. Transportations hazards are also there. So all these risks are required to be evaluated through subject matter experts having knowledge of the biological risk evaluation, production, and manufacturing of your devices. So one of the critical tasks also when in the implantable industry, and when we are saying my devices are safe. So what is happening after implantations? And some of the device are implanted in the combination of the different components. For example, bond plates and screw, hip systems, knee systems. So there are the combination of the different metals and polymers. So there may be chances of the different types of the corrosion or wear that may occur during the implantation, after implantation in the life cycle of the patients. So we had understood earlier, we had understood all these, that is before the commercialization of the devices. Now in some of the cases, and we also understood that is the life cycle of the device. In some of the cases, after implantations, as per claims, the possibility of the corrosion is also there. So there are different types of corrosion here mentioned, and that is mainly applicable to the orthopedic implants, and different types of corrosions may lead to your device depending on the nature of your device. It starts from the general corrosions to the general corrosions means that we are saying on our metal sets, that is the normal corrosions may appear on the device's surface. Active corrosions with the involvement of the active implants means those requires the involvement of the energy source for the functioning of the medicals. So the presence of corrosions due to the effect of the energy, electricity. For example, the endoscopes those are being inserted into the human colon required to be connected with the high radio frequency instrument generators. So the manufacturers, those who are direct only manufacturing the those electrodes, are saying generally yes, we don't manufacture the instrument. But during the clinical applications, your device is required to be connected with the high radio frequency instrument. So it's an active device, and you should also understood the possible risk of the corrosions and biological safety of your device in active conditions. Galvanic corrosions generally occurs when two different metals are in electrical contact. Means titanium to SS to chromium. So there are the different types of metals that is recommended for the manufacturing of the implants. So you should also identify the risk of the galvanic corrosions. Pitting corrosions are also there. So slides have some information of the standards to be followed depending on the nature of the corrosions. This all information is derived from the FDA guidance documents. So they are giving some ideas to follow the what are the tests depending on the nature of the corrosion. So the standard 10993 part 125 version talks on the risk management that we earlier discussed. It talks to conduct, and there is an annexure B given in standard which describe the methods to be followed. Whereas annexure C talks on the literature review, systematic clinical literature review. So you can find those all information by following those standard annexures. Again, when we are talking risk management, it must be as per ISO 14971. That is the basic standard for the risk management. And any effect of PMT, that is the physical parameters of the as per ISO 10993 part 19. Here it's mentioned post-market, and it's not a post-market, it's a physiology, metrology, and topology. So all this evaluation that we earlier talked, it should be conducted from the subject matter expert having multidisciplinary knowledge. So again and again, standard talks to involve for toxicologist for the toxicity concerns and safe toxicity. So there are different globally available toxicologists depending on the your requirement and worldwide recognitions. So once you involve or consult any toxicologist, at least you should ensure the credential of your toxicologist and your recognition, their recognitions. So US based, the DATB and ATS are the globally recognized. When you are talking for the Europe, you can also an involve toxicologist having a credential of the ERT. Japan and Asia is there. Recognition is good to that ensure the overall credentiality of the toxicologist. So the subject matter expert should have medical and clinical application knowledge, engineering and technical knowledge of the product design, and having the knowledge of the risk management to be followed for the biological evaluations. Now the part comes to the laboratory that is required to be involved for the chemical characterizations, physical characterizations, and conducting the biological effects. It means different tests that is identified by the either toxicologist or from tables. So the first preference as per standard is doing the biological equivalence. If it is not, if you are not following, then second part is required to conduct the some biological test. So you should also ensure the requirement to be verified from laboratory before assigning contract with them. So for India, MD40 license is mandatory, that is being controlled from the CDSU. And if you refer the database of list of testing that is available on the Sugam portal, if you search, so you should find the lots of the laboratory in India that is certified under MD40. Again, once you find the lab, you should also ensure the scope of your test you are conducting from them. So your laboratory must have those all the tests that you are conducting from them. So this is one of the example. It is not for the promoting any laboratory. That is the screenshot is taken from the official database of the Sugam medical device. So when you are conducting tests for the India, at least in India, so you should ensure your laboratory is certified under MD42 with this applicable scope. The next accreditation of laboratory is the NABL 17025. So NABL 17025 scope can be verified from the different database. So there is a one, if it is from the Indian accreditations, the database are available on these websites. You can, if they have some different international accreditation, you can verify the authenticity of the certificates along with the scope that is being conducted. When you are going for the European certification CE mark, so they are demanding having the NABL 17025 accreditations of your laboratory. Even in India, CDSO is also recommended to conduct the test having 17025. So now we go for the USA. If you are thinking to plan your device for the 510K submissions, so nowadays FDA, US FDA had recommended to at least follow the ASCA conformity assessment. So ASCA testing accredited laboratory. ASCA means accredited scheme for conformity assessment that is recognized from the US FDA. So they need to follow the GLP requirements of the US FDA. And as on today, and by referring the test on doing the search on 22nd June, I had found that these laboratories are found to be complying to the stage 1 of the ASCA. So that is the A2LS certification. That is the minimum step required for the accreditation from the ASCA list. So you should find in India there are these 6 labs I found during the search from the database of the A2LS certifications having different codification of the C1, C2 and C3 that is given on the website. So these all are the basic steps. These labs are ASCA not certified means they are A2LS certified. If you want to again verify the ASCA accreditations, the US FDA is also publishing one list. So if those who are ASCA certified from FDA means that is available on the FDA website only. A2LA is the primary step that is required to for the ASCA conformity. Once your lab is having ASCA conformity, then FDA is again conducting the audits on your laboratory. And they are, once you are complying the all the requirements of the FDA, they are enrolling your lab to in their list. So when you are thinking for the 510K or US region accreditation, you must assure the ASCA conformity. It is not mandatory to follow having the ASCA, but they are recommending. Why they are recommending? Because once you refer the one articles of the 2024, maybe in September or August, so they had identified the article is still present on the website, and you can they had literally mentioned the name of the two countries, including India and China, and they are saying that they had identified some fraudulent activity or fake reports generated from these countries. So they had started controlling this laboratory under ASCA scheme. Still, there mentions it is not mandatory to have ASCA accreditation for the laboratories, but they are recommending to have the ASCA for testing being promoted for the Europe. So some of the countries other than well developed that we talk on the Europe and USA, and South Africa and other countries are the part of the OECD, Economic Operators Unions, and they have developed some laboratory practice that is unique and harmonized for the worldwide to be followed by the laboratories. So if you are thinking for the other regions, at least you should ensure the laboratory is having the OECD certification, GL. For animal care, there are two certifications that are available and required to be verified from the laboratory is the AAA LAC and second one is CCACA. So these two accreditation refers to the good animal welfare being conducted and mentioned in the ISO 10993 part 2. So directly conducting testing at any laboratory without understanding regulatory requirement is also a problem. And it may lead to some over compliance or non compliance in coming days if you are following the other strategy. So at least you should ensure in understanding worst case scenario, you should conduct the test from laboratory having almost all the certifications. So it does not become any obstacles in coming days if you change any regulatory. So I said in earlier slide, the part 12 talks on the sampling, minimum sampling, and what are the ways of the samplings. And so it talks on the different extraction condition of your devices. Medical devices are extracted with the different solvents depending on the nature of your device, maybe either polar, non-polar, and other. So standard talks to follow the specific ratio in your samples must be available for the extraction that is being used for the testings. It depends on the quantity, that is surface area or weight by gram of your devices. So you should follow that K3 that is given in the table. Depending on the nature of your devices, and you should ensure the minimum quantity of your device is being used. So firstly, you should identify what are the minimum quantity required for the test. Then after, you should identify which temperatures condition is being used for the extractions. So they had given the different parameters of the extraction temperature and time. So your device is submerged in the different polar and non-polar solvents and other solvents for the specific quantity for a particular temperature and time, depending on the clinical use of your devices. The biggest problem that I had seen in industry in laboratory is that by doing the extraction, you should ensure what part is being extracted. When you are submitting a device, for example, infusion sets, so you should ensure that components of the device that are not coming in contact is not required to be submitted. For example, flow regulator of the devices. So that is not coming in contact with the patient in any way. Then why are you incorporating in the samples? Second thing, same scenario for the infusion sets. So the internal lumen of the device are mainly coming in contact with the patients. So people's or laboratories are directly cutting your tubes and doing the testings. My question, or I identified one risk is that you must do a extraction of the internal surface of your device only, irrespective of the external surface, because during manufacturing techniques, so many operators had touched the external surface of your devices. So there may be some other biological risk, but even they, that is not going to the patient. So false positive chances of false positive results are there. So you should also understand which part and inner, outer, whether where it is coming. So that is also a crucial part to understood for the extraction condition. So this is the extraction conditions as per 10993 part 12, whereas for the breathing gas pathway, it does not talks on the specific sample quantity to be tested. Whatever the samples you are submitting for the testing must be in the finished forms. If your device is sterile, you submit the device in sterile condition. If your device is intended to be sterilized during the application of your device, you recommend your laboratory to follow the same conditions that is mentioned in your IFU to claim the biological equivalence and to generate the simulation environment of your devices during the environment. The standard talks to conduct a different toxicological test. But when you are claiming your device is maximum including foreseeable misuse is clinically intended to be used up to the 10 days. So rather than going for the 13-day study, you can also reduce your study up to the 10 days also. So we had understood overall the biological nature of the device, what are the tests, how to conduct, what are the requirement of the subject matter expert, including toxicologist and laboratory. Now, by following the standards and three principles of the reduce, reforms, and removal of the animal testings, the standard again and repeatedly talks to follow the chemical characterization of your devices. So there is one standard, part 18 is there, and it talks to perform a certain test. I'm just sharing you in the another slide. So if you refer the different tables of the ISO 10993 part 18, it talks on the qualitative and quantitative methods looking to the different nature of your device, maybe in the polymer form, metal, ceramic, and other forms. So it gives idea what are the minimum tests to be conducted for the chemical characterizations. So when you involve any laboratory and toxicologist, At least you should understand what is the scope is minimum requirement of as per the standards, as per the category of your devices. So for polymers, it's a different strategy. For metal alloys, it's a different strategy. So by following these chemical characterizations, so they should come out with the different leachables and extrables that is found by following the specific extraction condition. So now the question comes, if something is identified in the chemical that is leaching, either leach or extractable from your devices, whether it's toxic or not. What are the quantities if it is present in your device? How much quantity is present in my devices? Whatever the quantity present, whether it's a safety relating to the biological safety of the patient or not. So for answering all those, there is a part 17 chapters and which talks on to identify the safety of margin. So the chemical characterization process is divided in 5 steps as per standards. The first step is you should have or you must gather all the information as described in these slides relating to the manufacturing process, materials. So before initiating activity, you must have some qualitative data of your raw material and manufacturing process and understanding the regulatory requirement and nature of your devices. So as mentioned in the biological equivalence, step 2 is for you to conduct the study and understand whether your material is equivalent to the predicate device or not. So follow doing this activity, you must have need to follow understood the some literature study and required to be understood depending on the nature of your devices, and you need to read and understood the lots of the literatures and their quantities and natures and clinical conditions and scenarios for doing the material equivalence. So for literatures, these all are the possible database and sources required to be reviewed and consult for getting the toxicological information. So again, you or me, even I am not the competent persons to do a toxicological evaluations, because once you identify the database and also collect the some literatures, uh, but depending on the limitations and we are unable to decode the what is mentioned in the literatures. So it involves the toxicologist to understand the overall requirement of your devices and what literatures are saying. So these are the all are the possible data source required to be followed as a literature identification relating to your medical devices. So after doing all this assessment, the toxicologist, the part 17 asks mainly to ensure the margin of safety, that is MOS of your devices. So what is chemically extracted and leachables, it must be in the margin of safety depending on the total tolerable intake or contact limit, that is TI or TCL. TI is quantitative whereas TCL is the surface, that is surface area depending on the nature of your device. So the margin of safety of your device must be greater than 1. If something is less than 1, then the detailed study is required to identify the biological safety of your devices. So once you refer and consult the toxicologist, generally they will talk with these different terms. And here I try to mention the all these terms and what are the definitions and how it is calculated. So though you consult the microbiologist, not toxicologist, you must have some knowledge of the part that is you are consulting or discussing with your toxicologist. So margin of safety is the ultimate character required to be declared from the toxicologist, and that gives the idea of the whether your material is safe or unsafe. Again, when you are conducting the chemical characterizations and your laboratory is saying that we have found the lots of the materials or substances that is possible or present and that is irrelevant to the your device, it means your manufacturing techniques is inappropriate. Again, you need to modify your design of the manufacturing. And you should have control on the manufacturing techniques, and you must remove those substances from your devices before conducting testing. So chemical characterization also gives idea of the what are the additional material that is going to the patient. By comparing the quality management system requirement of the ISO 13485, there is also one clause that is regarding the cleanliness of your devices. So additional substances that is going to your devices is also relating to the cleanliness of our devices. Directly or physical cleaning that is viewed by human eye is not the limited up to the cleanliness of your devices. So it does not mean it's a single time procedure of the chemical characterization. If something is not found or found in not favorable means some drastic change and then not irrelevant to your devices, you can change the design process, manufacturing process of your device also. You can again conduct and assure yes that by putting some controls, those are removed, and that is again verified from the characterization by identifying presence or either presence or either presence with the reduced manner or not. If you are unable to reduce and device is required to be presented in then form, if something is identified in chemical study, then it's a role of the toxicologist to understood the biological by doing the risk assessment. So the toxicologists require and they also need to follow the some limit that is defined in the ICH guidelines and ISO TS 21736 guidelines, depending on the longevity clinical application of your devices. So they will consider the one baseline of the quantity of your devices, and they will suggest the test to the laboratory by what are the manners of the testing to be followed and what are the limits to be followed for the particular extraction method and technical methods to be followed for the chemical characterization. It is mainly applicable for the technical methods that is being used for the chemical characterizations. So once you involve the toxicologist, they will ask the lots of the questions, and the possible outcome of the any substance that is being identified in chemical study and risk assessment required to be followed in the last two tabular formats. So for breathing gas pathway, these are the default limit given in standard to be followed depending on the application of your claimed application of your devices. So the last step of your chemical characterization is the overall clinical exposure of your substances, that is either leached or extracted from the devices. So the overall biocompatibility tests, and had already we discussed, these are the general grouping and summary of the different series of the test. Part 1, 2, and 12 are the core framework of the overall all the series of the biocompatibility test. Part 3, 4, 5, 6, and 10 are relating to the cytotoxicity, skin sensitization and irritations, hemocompatibility, and genotoxicity. These are the overall general tests that is recommended as a biological effect in standard along with the part 11 and 23. For leachables and chemical characterization, we should follow the part 17 and 18. And depending on the different natures, you must consult the different. These are the one slide that is prepared. Once you identify the test that is being conducted at laboratory, they have different methods under every test. So once you refer those standard, it talks on the different tests to be conducted for the specific biological effect. So choosing those tests depending your device is also a skill. So whatever the test that is given, they have some advantage or disadvantage. They have some limitations, and depending on the different applications, whether they are coming in with the neurospinals or vascular systems or so and so. So every test have some limitations. So why you selected this that test method, irrespective of other method, is also a questions required to be answered to the regulatory authorities. This is the table that is recommended by FDA, having some additional test requirement identified in round marks, along with the typical requirement of the ISO 10993 Part 18. FDA had in last 25 May 2026, they had recognized the latest version of the ISO 10993 Part 2025. However, they had mentioned that their earlier recognition of Part 18 is valid up to July 29. So they must ensure or they will accept the conformity with the older standard up to the July 29. After that, you must have remedies for the compliance of the 10993 Part 25. So here FDA had identified some additional tests as per the older standard. So when FDA had recognized means it does not mean it is mandatory to follow the older version. if you are able to follow when recognized means if possible, you can follow the latest versions. If not possible, FDA will accept the last version up to the deadlines only. So earlier we talked on the part 18, and when doing the biological equivalence, it also talks to involve the part 18 for the part 19 for the physiological, morphological, and topographical characteristics of your device, whether it is affecting to your intended claim or not. So whatever the methods followed by analytical laboratory must be validated in terms of the accuracy, precision, limit of detection, limit of quantification, linearity, range, and your test report must have all the informations. So the overall outcome of evaluation will be through the biological evaluation plan (BEP) and biological evaluation report having certain requirements that is already mentioned in the part of the latest standard. The plan must have the device information, material information, preclinical and toxicological data with processing aid information and some of the different suppliers, regulatory cross-reference requirement of the basic QMS of that is ISO 13485 risk management, generic standard. If you are using breathing gas pathway, you can also incorporate the suggested and dental. So you should follow the cross-reference all the regulatory requirements mentioned in your standards. Justification of why biological testings is required, minimum required. Why you are not follow the clinical characterizations. Who had conducted the evaluation, that is subject matter expertise and competency of those persons who had involved, and limitations of your evaluations. Limitation of our evaluation is very crucial to understand where we are limiting the overall evaluations. So limitation of your evaluation may be limited to the any changes in the source of the specification of raw material used in the manufacturing of the product. So if you are going to change your basic raw materials, that is subject to again new re-evaluation. A change in the formulation process, processing, manufacturing, other processes or primary packaging or sterilization methods also subject to a evaluation. So these all are the possible re-evaluation criteria, and these may all become the limitations of your biological evaluations. So it's not a single time process, because in routine manufacturing, we are going to change methods, raw materials, presentation styles, everything, but that is subject to a biological evaluation only. Thank you. It's closed. And if you have any questions, please ask.
Now I request all the delegates to unmute yourselves and ask questions. You may also type your questions in the chat box. So there is one question in the chat box from Akshita, that what happens if a device is not biocompatible?
If device is not biocompatible, whether it is identified at the chemical characterization stage or from the biological testing. If it is biocompatible, so you have to change the basic raw materials if it is not or identify the main cause of the why it is non-biocompatible. If your basic raw material is complying, so as said in the discussions, you had added something additional during the manufacturing process or in the packaging processes that is required to be nullified. You cannot sell and distribute non-biocompatible products in market. It must be in safe manner. That is the evaluation why we are performing for our devices.
So there is one more question from Hemendra. We are manufacturing implantable device. One raw material supplier has been changed, but the material grade remains the same. In this case, do we need to repeat biocompatibility testing as per ISO 10993?
So, in my up to best of my understanding, when you are assuring the basic grade of your materials, you don't need to perform the retesting. However, in your documentation, you must ensure. Okay, earlier, I was procuring this grade that is conforming to the ISO 5832-1. So earlier, I used to procure from the X manufacturer. Now I am procuring same grade from the Y manufacturer. So it does not raise any questions on the safety and efficacy of your devices. So I think you should only update the supplier information in your biological evaluation plan only. And come out with the judgment, it does not raise any concern of the biological safety. So you are continuing with the same material of the Y supplier. That is the one question when he had asked one question that is mentioned in the
So there is one more question from Hemendra sir. Is a supplier statement in the certificate of compliance for biocompatibility testing as per ISO 10993 sufficient, or do we need to conduct testing?
So whatever the supplier you had, whatever the reports that is you received from your supplier, again required to be evaluated as per the overall requirement of the standards. In many cases, the suppliers are providing some limited test results only. But we are not directly selling that those part of your it may be a part or constitute of the device of the any supplier's item that is we are integrating in our medical device. So here we are talking on the medical device in finished forms. So you might have added some manufacturing process and other parts in your devices. So overall evaluation must be done in the finished form of your devices, and you need to ensure whether is there any additional biological risk are generated during the manufacturing, packing, sale, distribution, and every part of the life cycle of the devices. So if all the questions is saying no, nothing is, no biological risk is generated, we are just only doing the packing and my packing is also safe. In that case, you can say yes, the supply test report is more than sufficient. If some questions are raised, so when in slides we had told that whatever the evaluation done must be from the subject matter expert, having some limitations of the involvement of the toxicologist, or having knowledge of the risk management process and having knowledge of the product manufacturing. So if you had involved the subject matter expert in realistic way, so it should give the exact idea whether is there any additional risk is generated or not. So there is no standard in the world. We talk the how we to identify the specific risk relating to the your suppliers and XYZ. So that is required thoughtful process and to be followed for risk management of the devices. Generally, biocompatibility evaluation of the supplier data serves as a supports the biological evaluations, but it's not the ultimate biological evaluation. You must conduct biological evaluation plan, and by doing the plan, there are possible outcome is biological further test is needed or not.
One more question from Nikhil, that nanomaterials, anything more required?
So there is a part 22 in 19002 series, and that is required to follow along with the involvement of the toxicologist to understand the nature of the nanomaterials and how it affects. When you are involving the nanomaterials, part 19 is also applicable.
Sundar sir, over to you.
Yeah. Thank you very much. Mr. Sheth, it was excellent. With a given limited time, you could cover almost every aspect of this biocompatibility. Our experience, I've been involved in this almost 30 years now. But our experience is there that in India, manufacturing take it very lightly, these requirements. Like this, I can give you example. We had been certifying condoms where you use silicone oil plus you use flavors and all those things. But then they say biocompatibility not required. And we didn't certify, but some other certification body certified them. So that's
Because
shortcuts are there. Like this, recently, orthopedic implant, especially knee joints and other joints which are critical joint, arm joint, hip joints. Now, they say biocompatibility not required. Their consultant, how they can say this? And we find it difficult to convince them. We reject, of course, but we find it difficult. Same thing endoscopy and even ECG. Now you are using gel that's coming directly contact and is coming in contact more than specified time. Lubricant used in this endoscopic or the diagnosis purpose. But then we say this is not acceptable. You have to have compatibility, biocompatibility. Whether they are not aware where this such facilities are available to the testing and clinical testing. Can you put a light on this? And that's a reason China and India seen with the red-eyed by your USA market.
So definitely, sir. So your question is very valuable, and this happens because India started involving GMP in 1996 only. The pharmaceutical manufacturers are not aware about the AHU, even air handling unit requirement in the manufacturing plants. So I think up to best of my knowledge, in more than 90% companies had started involving AHU as a GMP part since 1996. The medical device is controlled in India in 2017 and implemented in 1st January of 2028. Again, they had given the transition period for the existing license for the 5 years. So nowadays, some of the regulatory enforcement from the CDSU is good, and they are assuring the biocompatibility of the device. Even they started controlling laboratories by making separate provisions for the laboratory. That is, I mentioned in this slide for the laboratory accreditations for the India. So once you refer the Sugam medical device, so it gives the list of the medical testing laboratories. Those are accredited under MD40 license. And once you refer their license, they have list of all the tests that is mandatory or required to be performed under ISO 10993 part all the series. So when you are going to conduct the cytotoxicity with the laboratory having MD14 license, but they don't have this test in scope, so it's useless for you. So at least you should understand the scope of the testing laboratory and what test you are going to conduct. The recent, again and again saying the regulatory bodies are asking to reduce the animal testing by replacing it with the chemical characterizations only. Everything is safe because more than 90% devices are manufactured with the raw materials that is generally suggested with the implantable growth, and they have long and safe history in the industry. But the manufacturing techniques that we used to follow for the commercialization of device is very dangerous. And that is the main scope of the biocompatibility evaluation of the devices only. So since last 5 years, the demand is from the regulatory authorities had triggers the doors for the chemical characterizations only. And you can skip all the biocompatibility test by doing the systematic evaluations by involving the toxicologist. So depending other than the India, and we should also understood the conformity requirement from the USA. If you are trying to, we have some interest in the Europe regions, or so what are the minimum requirements of laboratory for different regulatory? That is also a key part to understood before directly conducting testing.
Yeah, that's correct. Even the European norms are also very strict, and people have to really follow it, especially when it comes to clinical testing and biomedical testing, biocompatibility testing. So thank you very much for the clarification.
Thank you.
Back to our friend Pranali.
Yes, sir. So one more question in the chat box from Swati ma'am. If the device such as NIBF cuff that comes in contact with body for less than minutes, so do we still need to perform the biocompatibility test for them?
Less than 1 minute?
Yes, sir.
So as per the definition of the standard, it comes under the brief contact device, and if it is contacting the skin only, so you can claim the clinical environment material safety by considering the clinical environment and main material of the devices with the safe history of the device. You should find the so many literatures relating to your devices that talks on the specific clinical application of your devices with those specified raw materials as a base materials. So you need to prove the biological safety of your basic raw material only.
So I see there is no questions anymore, sir. So we are going ahead. Before that, on behalf of ICS and all the participants, I would like to express our heartfelt thanks and sincere gratitude to you, Viral sir, for your insightful and highly informative presentation. Your in-depth knowledge, practical expertise, and clear explanation of the subject have provided the valuable insights and enhanced our understanding of the importance of biocompatibility in ensuring the safety and performance of medical devices. We truly appreciate your valuable time, support, and wonderful contribution. Thank you for making today's session engaging, informative, and memorable. Thank you once again, sir. We wish you continued success and extend our best wishes on behalf of the entire ICS team. Thank you so much, sir. So ICS is placed to provide you all its research and development, integrated management system, QMS, CMS, and OHSMS, excellence in education management, ISO made easy in Gujarati, Marathi, Telugu, and Hindi. Our recent publication, first one is Urja, Thermal, Oil and Gas, Nuclear. Second one is Jung Se Jung Tak, Fight with Rust, Corrosion Causes and its Mitigation. For reading any of the above books, please contact Ms. Sushma Kindalkar. Feedback form. Please register on www.sadgunsam.org and go in your login and give feedback. We are officially closing today's session. Before that, I would also like to thank all our participants for their valuable time and active participation. We hope you found this session informative and useful. Have a nice day.